So the first one was looking at the effect of different numbers of clusters on T-scan, and you should have found that as long as you stayed above about 6 clusters, T-scan works well, but below 6, it starts merging time points together and works even worse than it does normally. And the other exercises. Yeah, and adding additional eigenvectors shouldn't really help the resolution of the later time points. This is because there's just much bigger differences between the earlier time points and the late time points than there are between the different stages in the late time points. And then, yeah, different case shouldn't have had too much of a difference on slicer, but again, extreme values would cause it to break. And using different sets of genes for this data set shouldn't make very much difference because both the feature selection methods worked reasonably well, and there are very strong differences between the cell types in this data set. But if you have smaller differences, then all of these different parameters will have can have a large effect on your analysis. And we're going to move on to imputation.
Right. So now I'm going to talk a little bit about imputation, which is a new chapter for this time we're running the course. So, and it's a relatively novel. The two methods that are coming out that we're using here are relatively novel. So the idea here is sort of harks back to the problem that we mentioned several times of having all these zeros present in the expression matrix. And we know sort of from biological reason, from the biology and from our understanding of our experiments, that sort of there are really 3 reasons why these zeros could appear. So basically, number 1 is that it's actually a real zero. In this cell, this gene was not expressed at all, and hence the 0 is an accurate representation of the fact that the gene or the transcripts were absent. Another reason, which is sort of more problematic, is if the gene actually was expressed, so there were transcripts present, but for some reason those transcripts, we were unable to capture them, or they got lost when the cell was lysed, or for whatever reason, they fell out before we were sequencing, and hence we record a 0 even though the transcript was actually present. And the third reason is that we did capture the transcript properly, but when we sequenced, we didn't sequence deeply enough, so that basically we failed to get any reads from the transcript, and hence it appears to be a 0 there, even though it was actually present. And I think that the primary reason for why you want to be doing so, this you could obviously address by just sequencing more deeply, but this one is not going to help you no matter how deeply you're sequenced. So this is really the reason why people are trying to do imputation. So the idea behind imputation is basically that you try to use some sort of underlying model of what is going on to basically sort of fill out these zeros and try to sort of infer what is the true value that was present there, but that we didn't observe. So needless to say, this is quite a difficult challenge computationally. I think the main reason why it's so challenging is because we don't have a sort of a really good theoretical model of the transcriptome and what kind of distribution it should be following. So hence, this tends to be a very sort of data-driven exercise. So as I mentioned, there are 2 methods available. Magic is unfortunately only available for Python and MATLAB, but we can run Python from within R as we did before with SNN-click. This was the first method, and a few months later, also this year, SC-Impute was published, which is an R package. So we're going to look at SC-Impute first. So here we've really sort of done all the work for you. So all we do is that we load the DENG data set again, and then we just run SC impute with the standard parameters. And now we first look at the results. So here is the PCA plot of what you see after you have done the imputation. So you should flip back to chapter 4.2 and sort of see what has happened compared to the original data. So if you want to do that. So this was the old plot. Back is not working. Okay, I'm going here. And this is a new plot. So not too much of a difference, I would say. Is everyone in agreement with that conclusion? This is sort of kind of a qualitative view of it, because we're just looking at a sort of projected version of the data. So to do it a little bit more quantitatively, we can ask sort of how does the imputation affect any of the sort of downstream analysis, because imputation would be something that you would do very early on before you did, for example, clustering or pseudotime analysis. So in this case, we're going to be doing clustering. So what we do is that we just run SC3, because that's our favorite method, for the imputed data, and then we're going to compare that result to what happened when we ran SC3 with the original data. So as you can see here, for the estimation of K, we still get 6 clusters. So in order to be able to make the comparison with the original data more easily, we still use K equals 10. And when we run it, what we notice is that the ARI that we got for this new clustering is actually significantly worse than what we had before when we didn't do imputation. And this is, we can sort of see why this is. We can't see it anymore because we don't have the similarity matrix. But if you do plot the similarity matrix, you will notice what has happened to the to it because of this imputation. And you can also sort of see by comparing this PCA plot where the clusters have been colored with the previous one where you had the original labels. You can sort of see what it has done to the clustering and where it seems to be mixing things up. Right. So before we do this exercise, we can just sort of show that we're doing the same thing with magic. So we're running this Python command, and then we're doing the PCA plot. And what we can see here is that we have something which almost looks like more like a trajectory than a clustering. So basically, and so what has happened here, especially if you look at the first component here, is that that the magic has for some reason almost put all the variance into the first principal component, and sort of turned this into almost a univariate data set where everything seems to be sitting on one line through this high-dimensional space. So this is obviously not good, because it seems to be sort of have destroyed a lot of the variability that was present in the original data. And this is sort of reflected when we look at the downstream analysis, where we sort of seem to be getting 4 clusters instead of 10, which is, or we got 6 before, we would have liked to seen 10, so that's obviously worse. And the ARI, when we compare the clustering, is also poorer. So so this is clearly not very useful for doing the. So I sort of already given away the answer here to the exercise, which is whether or not which one of these SC impute and magic you would recommend, or if any one of them at all, and if so, why. So I'll give you a few minutes to think about that. before we move on to the next session, next section here. Questions? Yes. No, I don't think so. I think these methods, they only look at the expression matrix and try to sort of, I mean, essentially what they do is that they do sort of smoothing based on the sort of of the other elements present in the row and column of where the 0 is found. But I do seem to recall that at least in the SC Impute paper, they point out that it would be a good idea to take these things into consideration, but I don't think the methods do that at this point. So, I mean, I don't know the sort of underlying machinery in these. How these methods work, and I haven't looked at the outputs in particular detail. But I think you would hope that if a gene is not expressed in any of the cells, that it should remain unexpressed in all of the cells, because you have no evidence to support that it should be expressed, right? The question is if these methods take the cell type into account. I don't think they do. I think they only look at the expression, because again, the idea is to use this prior to clustering, so they don't leverage that information. Yes.
So do you recommend that you do the imputation before the quality control or after?
So when to do the imputation, before or after the quality control? I think that you should do it after you've done the quality control. Yes, so I mean, presumably sort of, the reason you do quality control is that you assume that sort of all the poor cells will sort of have a negative impact on any downstream analysis. You want to get rid of them as soon as possible. So that's, yes? So the question is if the imputation methods have some sort of quality metric. Not that I know of. I don't think they have that. I mean, again, I don't think it's really possible to do, because it really depends on sort of how can you assess the quality of an expression matrix, right? It's not really possible, because it depends on how you define quality. So I don't think they have that. Okay, I think I'm going to hand this back to Tallulah, right?
All right, so now we're going to look at differential expression analysis. So for bulk RNA-seq, this is sort of the number one thing you'd be doing after you've got your data. So then you've got two or more biological conditions, and you want to find the differentially expressed genes between them. And there's lots of methods that have been developed for doing that for bulk RNA-seq. Probably the two most popular are Day2 and AJAR. And there's also been extensive testing of that, the performance of those methods. However, for single-cell RNA-seq, this often isn't sort of the case we have. Often we don't have two defined experimental conditions that we want to compare, and even if we do, we might have different cell types in those different conditions. So just comparing all of the cells in one condition to all of the cells in another condition might not make very much sense. So often we'll first be doing unsupervised clustering to identify different groups of cells, and then trying to find those genes that are differentially expressed between those groups to try and interpret biologically what those groups represent. When you're running SC3 in the previous chapter, it does the sort of differential expression between clusters using the Kruskal-Wallis test. However, unlike bulk RNA-seq, for single-cell RNA-seq, we have a large number of samples, which means we have more options other than just comparing the mean of the expression in two different groups of cells. We have a whole distribution of values, so we could test for differences in variability or other sorts of differences in distribution, rather than just differences in the mean expression. However, to compare distributions, to evaluate whether there's a difference in variability or something, we would have to fit a particular statistical model or distribution to the single-cell data. The other option is, if we don't want to fit some sort of parametric model, we could use some sort of non-parametric test to look for differences in distribution or differences in mean or median in our data. And this has the advantage of the fact that it doesn't matter if our model fits well or not, because we're using a non-parametric test. It doesn't matter what the distribution is. So these are some of the models that have been used to fit and describe single-cell RNA-seq data. So the most common model is the negative binomial model, which is part of R. It has two parameters, the mean and the dispersion parameter called size, which is related to the variance. And you can just plot what that looks like for different parameters. And so this is how the size is related to the variance of this distribution. However, the raw negative binomial model in general doesn't fit single-cell RNA-seq data very well, because we have problems with detection rate. We have a large number of zeros. This isn't captured by the generic negative binomial model that's used by EdgeR and DE-seq. So to account for this, people have developed a zero-inflated negative binomial model. This is used by the most common single-cell RNA-seq specific differential expression methods. So here you just have the mean, the dispersion, and a dropout rate. And you just assume that for every cell, there's some probability that it's going to be a zero just from the dropout rate. And then if it's not a zero, then it will be drawn from a negative binomial distribution. And you can again see what that looks like. And this looks a lot closer to single-cell RNA-seq data. And this is how you can relate the mean and the variance to these various parameters. However, the model that makes most biological sense and that there's the most experimental support for is actually a Poisson beta distribution. And this has three parameters, alpha, beta, and gamma. So I've called them A, B, and G here. So alpha is the probability of the promoter being turned on. Beta is the probability of the promoter turning off. And gamma represents the rate of transcription from that promoter while it's on. And if you follow that model for stochastic expression from a promoter, then you get a Poisson beta distribution. So first we draw a beta random variable using alpha and beta, and then we draw a Poisson variable using the mean, which uses the gamma and the lambda drawn from the beta distribution. And this is what that looks like, which looks the closest to what single-cell RNA-seq data usually looks like. And you can see this sort of long flat tail, which is not seen in the zero-inflated negative binomial model. Yeah, so then you can explore those different parameters for the different models. However, you should see that the equations for the mean and the variance are getting quite complicated for the Poisson beta distribution. And overall, it's much more difficult to fit the Poisson beta distribution to data. than it is for a zero-inflated negative binomial model. As a result, there's only a couple methods that use it for differential expression, and they're very slow and time-consuming. Most methods will use the zero-inflated negative binomial instead. So we're going to do an example of how to do this with actual methods that are available. So we're going to continue using the Blischak data, the Tung data, because along with the single-cell RNA-seq data, they also generated bulk RNA-seq data for the same cell lines from each individual. And we've pre-computed the differentially expressed genes from the bulk data for one pair of individuals that we can use as the ground truth to test different single-cell RNA-seq methods. So here we're going to define the ground truth, and then read in the Tung data again. And here we only want to select these two individuals because that's who we've got the bulk data for. The first test we're going to consider is the Kolmogorov-Smirnov test, which is a very simple non-parametric test. It's often referred to as the KS test, and it quantifies the distance between the cumulative distribution of expression for each gene in the two populations. So here, this red distribution would be the expression in one of your cell populations. This blue distribution would be the expression in the other population, and then it just adds the difference between these two distributions across the entire range of expression levels, which means it can pick up all kinds of differences, including an overall magnitude shift. So if the mean shifted, one of these curves would just be completely to the left or the right of the other one, but also differences in variance, which would cause one curve to be sort of more up here and then drop down to below the other one. This test could also pick that up. And it's just a basic built-in function for R, so we can easily apply this test using the apply function. So here, this is just norm is our normalized expression matrix. 1 indicates that we want to run this over each of the rows. And we just run the KS test with the group for group 1 as one individual and the other group for the other individual. And then take the p-value from that test. And we, of course, need to adjust for multiple testing because we're testing all 10,000 genes. Now we want to evaluate the accuracy of this test using the ground truth from the bulk data. So here we can look at how many genes it's identified as significantly differentially expressed. And you can see that it's over 5,000, which is a lot. It's probably incorrect. This is because it looks at the full distribution of expression values. That means the test is extremely sensitive to any sort of difference. So any slight difference in the distribution will be picked up by this test, but that also means you're going to find a whole ton of differentially expressed genes. So if we look at how many of the ground truth differentially expressed genes are found as significant, we can find that there's almost 800. But if we look at how many that we know are not differentially expressed, there's 3,000 that are false positives in this test. You can see that there's many more false positives than there are true positives in there, but there's also a lot fewer known to be truly differentially expressed genes than not differentially expressed genes. So we want to quantify this in a more relativistic terms. So we're going to look at the true positive rate, which is just the number of true positives. So it's the number of really differentially expressed genes that our test identified as differentially expressed, divided by the total number of truly differentially expressed genes. So that's the true positives plus the false negatives. So that's the genes that are differentially expressed that we missed with our test. And the false positive rate. So this is the proportion of genes that we identified as differentially expressed with our test that are incorrect, that are actually not differentially expressed. So that's we've got the false positives divided by the total false positives plus the true positives. Yep.
Why do we relate to the bulk? Maybe it's just like a silly question. Why do we relate to the bulk parameters? I mean, maybe we have a higher sensitivity in this one.
It's possible we have a higher sensitivity, but for this particular data set, these are homogeneous IPS lines being grown in culture. So we expect them to be the same. Obviously, for a real data set, like from a tissue or something, this would make no sense, because you wouldn't expect the bulk to capture the same thing as the single-cell RNA-seq. This is only for this particular data set that we can actually use it as a test case. Most of the time, you can't. So here's how you calculate those things. And see, we actually have a true positive rate of 70% and a false positive rate of 30%. So you can see that the true positive rate is much higher than the false positive rate, which indicates our test is identifying differentially expressed genes. It's just not all that great at it. However, so far we've only considered a single significance threshold. So we're only considering p less than 0.05 after multiple testing correction. It can be useful to consider different thresholds. So if we made our test more stringent, maybe the KS test would work a lot better. In order to summarize the accuracy across all the possible thresholds we could choose, we're going to use the receiver operating characteristic curve. This is the ROC curve, and we're going to calculate the area under that curve. So this is again how we do that. We have to first select just those genes where we know whether they are or aren't differentially expressed. There's a lot of genes who are ambiguous. We create our truth, whether it is or isn't differentially expressed, and we run the ROC package to create the area under the curve. So this is the receiver operating curve. So this is the false positive rate through positive rate as we vary the significance threshold. So if our test didn't work at all and we were just selecting genes by random chance, there should be a diagonal line from 0 to 1, 1. And we have an AUC of 0.5. So here's how we'll calculate the area under that curve for us. We're at almost 0.8. So it's quite a lot better than random chance. And here we're just going to save all that code into a function, so we don't need to repeat it for each new test we consider. The next test we're going to consider is the Wilcox-Mann-Whitney U test. So this is just a rank sum test. It's another parametric test, but specifically tests whether the values in one group are larger on average than the other group. Only does this using ranks, and tests whether the median expression is higher or lower in the other two groups rather than the mean expression, which means it's much more robust to outliers. And unlike the KS test, it shouldn't be picking up difference in variance or other differences in the distribution. So this is how you apply that test to our data, and we can calculate the quality again. This is the AUC curve. And you see we get an overall AUC of 0.83. So this is working better than the KS test. You can also use EDGAR, which was designed for bulk RNA-seq and is based on just a plain negative binomial. Doesn't make any account for the extra dropouts. However, it uses a general linear model framework, so we can include other factors beyond our biological conditions. So if you had different batches or different replicates, you can include those in the model, and they would be automatically accounted for. And then you wouldn't have to do batch correction prior to running the method. So we just have to create the object with our raw counts this time, and then calculate the size factors for this, and then the group identity for each of the cells. And then we create our model design. So here we're not accounting for batch effects. We're just going to look at the effect of the individual. Then we estimate the dispersions, fit the general model, perform the test, and then we extract the p-values of that test for a two-parameter, two-group test. And then we can extract the p-values and run our quality function again. So this is the results for EDGAR. And we get an overall AUC of 0.85. So even though it's using a model that doesn't really fit single-cell RNA-seq data very well, it actually performs quite well on this data. Next, we're going to look at monocle. So monocle uses a specialized negative binomial model that accounts for sequencing depth. For count data, you can also use log-transformed normalized data if you assign it a different model called Gaussian F to tell it to use a normal distribution to fit the data rather than a negative binomial. So this again uses the GLM framework. However, it requires batches to not be confounded with your experimental groups, which isn't the case for our data, so we can't use the batch information. And this is how you run it. So we already saw how we have to create our own object for monocle, and then estimate the size factors and estimate the dispersions, and we can use the differential gene test and provide our model as a string. And we take the results and plot the quality measures. So here's our AUC curve, our ROC curve. Again, and we get an overall AUC of 0.83 or 825. So even though this is now a model specifically tailored for single-cell RNA-seq, it's not outperforming the bulk methods. So I have an exercise here to compare the results from using the negative binomial on the counts, from using a Gaussian model on log-transformed normalized counts for that data, for this data. So I'll give you 10 minutes to work on that.